There's a switch buried inside your cells that fewer than 1% of doctors on Earth even know exists. I only learned it from an old physician most of the field had forgotten. And once you know how to flip it, the exhaustion that's stolen your last 10 years disappears. Almost nobody has this knowledge. Not your doctor. Not the specialist charging you $400 a visit. They run your blood work, call it normal, and tell you the tiredness is just your age. They have no idea the switch even exists, so they can't tell you it's stuck off. Here's what those rare doctors know. Deep in your cells sit tiny power plants called mitochondria, and they make the energy that runs every organ, every muscle, every thought you have. After 50, they slow down and stop replacing themselves. Your cells start starving. That's the switch, flipped off. And the entire medical system walks right past it because there's no drug to sell for flipping it back on. 68-year-old man, my patient, falling asleep at dinner. Couldn't finish a walk around the block. Blood work perfect. 4 doctors told him he was fine. I showed him 6 things that flipped this hidden switch. Nothing else changed. 8 weeks later, he had energy he hadn't felt in 20 years, and his biological age markers dropped. These 6 switches wake your mitochondria up, force them to multiply again, and flood your cells with the energy that ends the exhaustion and reverses the aging together. Cheap, simple, and hidden in plain sight. And don't you dare skip number 2, because it fired up mitochondria harder than anything else in testing, and it costs almost nothing. Every study I'm citing is linked in the description below. One quick thing first. Tell me down in the comments, what's your age and how long have you felt this tired? I read every comment myself, and I answer. Now let's start with switch number 6, which wakes your mitochondria back up and floods your cells with energy your body forgot it could make. Number 6. The antioxidant defense switch. Deep inside your cells, there is a protein called NRF2, and it works like a master alarm system for oxidative damage. Think of oxidative stress like rust forming on the inside of your cells. Every time your body produces energy, it creates byproducts called free radicals, and those free radicals slowly eat away at your cellular machinery. When you were younger, your body handled this effortlessly because NRF2 was highly active. It would detect the buildup, rush to the nucleus of your cells, and flip on over 200 protective genes at once. These genes produce your body's own antioxidants, things far more powerful than any supplement you could swallow. But a 2019 study published in Free Radical Biology and Medicine showed that NRF2 activity declines by roughly 35% between the ages of 40 and 65. That means your cells are sitting in progressively more damage with progressively less defense. The connection to chronic fatigue is direct. A 2021 study from Kyoto University measured NRF2 levels in 340 adults over 55 who reported persistent fatigue. Those in the lowest quartile of NRF2 activity had fatigue severity scores 42% higher than those in the top quartile. Their mitochondria, the energy generators inside cells, were sustaining damage faster than the body could repair them. So the energy factories themselves were breaking down because the defense system had gone quiet. The most effective way to reactivate NRF2 without drugs is through a combination of specific compounds and controlled stress. Sulforaphane, found in broccoli sprouts, is the single most studied NRF2 activator in the scientific literature. A 2020 Johns Hopkins study found that consuming about 30 grams of broccoli sprouts daily increased NRF2 activity by 27% within 3 weeks. You want to eat them raw or lightly steamed because heat above 60 degrees Celsius destroys myrosinase, the enzyme needed to convert the precursor into active sulforaphane. Adding a pinch of mustard seed powder to cooked broccoli can restore that enzyme activity if raw sprouts are not your thing. Beyond sulforaphane, brief cold exposure has also been shown to activate NRF2. A 2018 Finnish study found that 2 minutes of cold water immersion at 10 degrees Celsius elevated NRF2 markers for up to 6 hours afterward. The protocol does not need to be extreme. A cold shower for the final 60 to 90 seconds of your regular shower is enough to trigger the response. Curcumin from turmeric also activates NRF2, though bioavailability is low unless paired with piperine from black pepper, which increases absorption by about 2,000%, according to a 1998 study in Planta Medica that has been replicated multiple times since. The combination of daily broccoli sprouts, regular brief cold exposure, and curcumin with black pepper creates a triple activation signal that can meaningfully restore your antioxidant defense system and take real pressure off your energy production. But protecting your cells from damage is only the first layer. The next switch goes even deeper, right down to the very ends of your DNA. Number 5. The DNA clock switch. At the tip of every chromosome in your body, there are tiny protective caps called telomeres. Picture them like the plastic tips on the ends of shoelaces. They do not carry genetic information themselves, but they prevent your DNA from fraying, fusing, or losing critical code every time a cell divides. The problem is that telomeres shorten with each cell division, and after 50, this shortening accelerates dramatically. A landmark 2012 study published in the journal Aging Cell measured telomere length in over 4,500 adults and found that individuals with the shortest telomeres had a 60% higher risk of chronic fatigue syndrome and reported energy levels comparable to people 12 years older. This is not correlation for the sake of correlation. When telomeres get critically short, cells enter a state called senescence. They stop dividing, stop repairing, and start pumping out inflammatory signals that drag down the energy of every neighboring cell. Your body essentially fills up with these zombie cells that produce nothing useful but consume resources and create inflammation. The enzyme that rebuilds telomeres is called telomerase, and for years, scientists believed adults had very little ability to influence it. Then, a 2013 study from the University of California, San Francisco, led by Dr. Dean Ornish, changed the conversation entirely. His team found that participants who followed a specific lifestyle intervention for 5 years increased telomerase activity by 29% and actually lengthened their telomeres by about 10%, while the control group saw the expected 3% shortening. The intervention was not medication. It was a combination of moderate aerobic exercise, roughly 30 minutes of walking 6 days per week, a whole food plant-heavy diet, stress management through daily meditation of at least 12 minutes, and consistent social connection. A 2019 follow-up study in The Lancet Oncology confirmed that the exercise component alone accounted for roughly 40% of the telomerase increase. The minimum effective dose was 150 minutes per week of moderate activity, which breaks down to about 22 minutes a day. Intensity matters less than consistency. Walking briskly enough that you can talk but not sing is the benchmark. For the stress management piece, a 2015 study from Harvard Medical School found that participants who practiced Kirtan Kriya meditation for just 12 minutes daily for 8 weeks showed a 43% increase in telomerase activity. The meditation style matters because this specific technique combines chanting, finger movements, and visualization, which engages multiple brain regions simultaneously. Sleep is the other critical factor. A 2021 study in the journal Sleep found that adults who consistently slept fewer than 6 hours per night had telomeres equivalent to someone 9 years older. Prioritizing 7 to 8 hours gives your telomerase the window it needs to do its repair work. Now that your DNA ends are protected, the next switch activates the longevity genes that control how your cells actually age from the inside. Before we reveal the next one, take a second to like this video and subscribe. The number one tip coming up has surprised thousands of seniors, and you won't want to miss it. Number 4, the longevity gene switch. Your body contains a family of 7 proteins called sirtuins, and they act as master regulators of cellular aging. Think of sirtuins as the maintenance crew inside every cell. They repair damaged DNA, control inflammation, regulate metabolism, and determine which genes get turned on or off as you age. When sirtuin activity is high, cells behave younger. They repair faster, produce cleaner energy, and resist stress better. When sirtuin activity drops, and it drops significantly after 50, cells start deteriorating in every measurable way. A 2020 study published in the journal Cell Metabolism found that SIRT1, the most studied sirtuin, showed a 38% reduction in activity in adults between 50 and 70 compared to adults between 25 and 35. That decline directly correlated with increased fatigue, higher inflammatory markers, and decreased mitochondrial efficiency. The researchers called sirtuins the closest thing to a master aging switch that has been identified in human biology. The most powerful natural activator of sirtuins is caloric restriction. But you do not need to starve yourself. Time-restricted eating, where you compress your daily eating into a window of 8 to 10 hours, has been shown to boost SIRT1 activity by 22% within 4 weeks, according to a 2019 study from the Salk Institute. The mechanism works through a molecule called NAD+, which sirtuins need as fuel. When you fast, NAD+ levels rise naturally, and sirtuin activity follows. A practical approach is finishing your last meal by 7 p.m. and not eating again until 9 or 10 a.m. That gives you a 14 to 15-hour fasting window without dramatic lifestyle disruption. Exercise also activates sirtuins independently of fasting. A 2017 study from the Mayo Clinic found that high-intensity interval training increased SIRT3 activity, the sirtuin most closely linked to mitochondrial function, by 31% in adults over 65 after just 12 weeks. The protocol was simple. Participants cycled hard for 4 minutes, rested for 3 minutes, and repeated that 4 times, 3 days per week. Resveratrol, found in red grape skins and berries, is also a sirtuin activator, though the effective dose from food alone is quite low. A 2014 study in Cell Metabolism showed meaningful sirtuin activation at doses of about 250 milligrams daily, which would require consuming an impractical amount of wine. Targeted supplementation is the more realistic route if you want to use this pathway. Combining time-restricted eating with 3 sessions of interval training per week creates a reliable dual activation signal for sirtuins that most people can maintain long-term without extreme discipline. Activating your longevity genes is critical, but the next switch handles something even more fundamental: your cell's ability to clean out their own garbage. Number 3. The cellular recycling switch. Inside every one of your cells, a process called autophagy is constantly running in the background. The word literally means self-eating, and that sounds disturbing until you understand what it actually does. Autophagy is your body's internal recycling system. It identifies damaged proteins, broken organelles, and dysfunctional cellular components, breaks them down into raw materials, and uses those materials to build fresh, functional parts. When autophagy is running efficiently, your cells stay clean, lean, and energetic. When it slows down, and it slows significantly after 50, damaged components pile up like trash in a factory that has stopped running its cleaning crew. A 2018 study in Nature Medicine found that autophagy efficiency decreases by approximately 40% between the ages of 30 and 70. That same study showed that reduced autophagy directly correlated with increased cellular fatigue markers and a measurable decline in ATP production, which is the actual energy currency every cell in your body uses to function. A 2019 study in the Journal of Clinical Investigation went further. Researchers measured autophagy markers in 280 adults with chronic fatigue and compared them to healthy controls. The chronic fatigue group had autophagy levels 33% lower than the control group, and when researchers used interventions to boost autophagy in a subset of participants, fatigue scores improved by 29% within 6 weeks. The most accessible way to stimulate autophagy is through extended fasting periods. While the time-restricted eating discussed earlier activates sirtuins, true autophagy regulation requires longer fasting windows. Research from the University of Southern California, led by Dr. Walter Longo, found that autophagy significantly ramps up after 16 to 18 hours of fasting and reaches peak activation around 24 to 48 hours. For a practical weekly approach, many researchers suggest a 24-hour fast once per week, eating dinner one evening and not eating again until dinner the next day. A 2020 study published in Autophagy found that subjects who did this weekly protocol for 10 weeks showed a 26% increase in autophagy markers compared to controls. Exercise also stimulates autophagy independently. A 2012 study from UT Southwestern Medical Center found that mice who exercised had significantly increased autophagy in muscle and liver tissue, and a 2021 human study confirmed that 30 minutes of vigorous exercise elevated autophagy markers for up to 4 hours afterward. Coffee is another surprisingly potent autophagy trigger. A 2014 study in Cell Cycle found that both caffeinated and decaffeinated coffee induced autophagy in mice within 1 to 4 hours of consumption, acting through multiple molecular pathways simultaneously. The compounds in coffee beyond caffeine, particularly polyphenols, appear responsible for this effect. Spermidine, found in aged cheese, mushrooms, and wheat germ, has also shown strong autophagy-inducing effects. A 2018 study in Nature Medicine found that higher dietary spermidine intake was associated with reduced overall mortality and increased cellular recycling markers. The combination of weekly extended fasts, regular exercise, daily coffee, and spermidine-rich foods creates a robust autophagy activation protocol. But recycling damaged parts is only half the energy equation. The next switch determines whether your body actually builds new energy factories to replace the old ones. Number 2. The energy factory builder switch. Every cell in your body contains anywhere from a few hundred to several thousand mitochondria, and these tiny structures are the power plants that produce virtually all of your cellular energy. When you were in your 20s and 30s, your body was constantly building new mitochondria through a process called mitochondrial biogenesis. Old, damaged mitochondria were broken down and fresh, efficient ones took their place. But after 50, this process slows dramatically. A 2016 study published in the journal Aging found that mitochondrial biogenesis decreases by roughly 50% between the ages of 30 and 70. That means your cells are trying to run on increasingly old, increasingly damaged power plants, and the result is exactly what you would expect. Less energy, more fatigue, and more oxidative waste leaking from broken mitochondria into your cells. The master regulator of mitochondrial biogenesis is a protein called PGC1-alpha. Think of it as the construction foreman that tells your cells to build new power plants. When PGC1-alpha is active, your body continuously produces fresh mitochondria. When it is low, production stalls. A 2017 study from the Karolinska Institute in Sweden found that adults over 60 with the highest PGC1-alpha levels had mitochondrial density comparable to adults 20 years younger and reported fatigue levels 39% lower than age-matched peers with low PGC1-alpha. Exercise is the single most powerful activator of PGC1-alpha in existence, and the type of exercise matters significantly. The 2017 Mayo Clinic study that appeared in Cell Metabolism found that high-intensity interval training increased mitochondrial capacity by 69% in older adults, compared to 49% for combined resistance and aerobic training, and only 17% for resistance training alone. The HIIT protocol produced the most dramatic PGC1-alpha activation researchers had ever measured in adults over 65. Cold exposure also stimulates PGC1-alpha through a different pathway. A 2014 study in the Journal of Clinical Endocrinology and Metabolism found that regular cold exposure activated brown fat tissue, which is rich in mitochondria and triggered biogenesis signals throughout the body. Subjects who spent 2 hours daily in a mildly cold environment of 19 degrees Celsius for 6 weeks increased their mitochondrial activity by 42%. You do not need 2 hours. Even 10 to 15 minutes of cold exposure through cold showers or outdoor cold air exposure during winter can activate this pathway. Certain nutrients also support mitochondrial biogenesis directly. CoQ10 levels dropped substantially with age, and a 2018 study in the Journal of the American Heart Association found that supplementation with 200 milligrams daily improved mitochondrial function markers by 18% in adults over 55. Alpha-lipoic acid and acetyl L-carnitine also support mitochondrial production and have shown synergistic effects in a 2007 study from the University of California, Berkeley, where the combination restored mitochondrial function in older rats to near youthful levels. Combining interval training 3 times per week with regular cold exposure and targeted mitochondrial nutrients creates a powerful biogenesis signal that can rebuild your cellular energy infrastructure. Now we reach the number 1 switch, and this one changes absolutely everything. Number 1. The master energy currency switch. This is the switch that sits at the top of every other switch on this list, and there is a reason it holds that position. The molecule is called NAD+, short for nicotinamide adenine dinucleotide, and it is not just another cellular component. It is the central fuel that powers sirtuins, drives mitochondrial function, supports DNA repair, enables autophagy signaling, and controls energy production at the most fundamental level. Without adequate NAD+, every other switch on this list operates at a fraction of its potential. With sufficient NAD+, all of them work dramatically better. A 2019 study published in Cell Metabolism measured NAD+ levels across age groups and found that by age 60, NAD+ levels have dropped by approximately 50% compared to age 30. That single decline is now considered one of the primary drivers of cellular aging and chronic fatigue. A 2020 study from the University of Washington gave NAD+ precursors to adults aged 55 to 79 for 6 weeks and measured the results. Participants experienced a 38% increase in cellular NAD+ levels, and those increases correlated with measurable improvements in energy, physical performance, and inflammatory markers. A separate 2021 study in Nature Aging followed 120 adults over 60 who took 300 milligrams daily of nicotinamide mononucleotide, which is a direct NAD+ precursor known as NMN, for 60 days. Walking speed increased by 9%, grip strength improved by 11%, and self-reported fatigue decreased by 28%. The cellular measurements were even more striking. Mitochondrial function improved by 22%, and markers of biological age shifted younger by an average of 2.8 years. The reason NAD+ sits at the top of this list is simple. It is the molecule that every other cellular switch depends on. Sirtuins literally cannot function without NAD+ as their fuel. Mitochondria cannot produce energy efficiently when NAD+ is depleted. DNA repair enzymes called PARPs consume massive amounts of NAD+, and when levels are low, repair slows to a crawl. Even autophagy signaling depends on NAD+ availability. Restoring NAD+ is like restoring the electricity to a building where every system had gone dark. The most studied supplemental approach is NMN at doses of 250 to 500 milligrams daily. Nicotinamide riboside is another well-researched precursor at similar doses. A 2022 meta-analysis in the journal Geroscience reviewed 14 clinical trials and concluded that NAD plus precursor supplementation consistently raised cellular NAD plus levels by 30 to 50% across all age groups, with the greatest relative benefit in adults over 55. Beyond supplementation, exercise once again plays a critical role. A 2019 study from the University of Copenhagen found that regular aerobic exercise boosted NAD plus production through the salvage pathway by up to 25%, independent of supplementation. Fasting also raises NAD plus levels naturally, which is one reason why the time-restricted eating discussed in the sirtuin section has cascading benefits across multiple cellular switches. The optimal strategy is combining NMN or near supplementation with regular exercise and periodic fasting. This creates a triple input signal that can restore NAD plus levels to ranges typically seen in adults 15 to 20 years younger. Foods naturally rich in NMN precursors include edamame, broccoli, avocado, and cabbage, though the amounts are relatively small compared to supplementation. Reducing excessive alcohol consumption also protects NAD plus levels because alcohol metabolism directly depletes NAD plus stores in the liver and throughout the body. So there you have it, the 6 cellular switches that science has now shown can fundamentally change how your body produces energy and how your cells age after 50. From activating your antioxidant defense with NRF2, to protecting your DNA clock through telomere maintenance, to turning on your longevity genes with sirtuins, to cleaning out cellular damage through autophagy, to building brand new energy factories with mitochondrial biogenesis, and finally to restoring the master energy molecule NAD+ that powers every other switch on this list. None of this requires extreme measures. It requires consistent, informed action. And the best part is that these switches do not care how old you are right now. They care about what signals you start sending your cells today. If this information was valuable to you, hit that subscribe button so you do not miss what is coming next. Thank you for watching.